Our second speaker will be Dr. Jane Wu, and I'm very happy to have her on this panel. And Dr. Jane Wu is a professor of neurology and psychiatry. Her group has been studying the molecular pathogenesis of ALS and FTLD together, both in the molecular biology, genetics, and model systems. And recent studies from her group and others have uncovered the link between nuclear RNA binding proteins and mitochondrial dysfunction in ALS FTLD spectrum disorders. So today she will discuss about recent advances in our understanding of the molecular mechanisms underlying the mitochondrial impairment in ALS and the implications in developing therapeutic approaches for ALS. So Dr. Jane Wu has been elected to the Fellow of the American Association for the Advancement of Science. And I can actually talk maybe days about her because she was the person who walked me to my hotel when I first came to Northwestern and for my recruitment dinner. And she was the person who convinced me to come to Northwestern. And she has been a role model, not to me, but to many women scientists. And I know because the meeting is via Zoom, there will be many children who are also watching this with their parents, with their grandparents. And especially for young girls, I would like them to pay attention to Dr. Jane Wu's talk because she's a true scientist and researcher. Nothing is more important than her science and research. And I am very happy to have known her and worked with her in the past. So I usually ask people if they could send me a childhood photo. It was very hard to get one from Dr. Wu, but I want to tell you this, that she actually worked together with her daughter and produced a movie. And that movie was acknowledged or even suggested for an award. And I think she has multiple sides to her, not just as a scientist, but as a mother, as an artist, as a film producer, and a very adamant supporter of girls in science as well. So it's my honor to have Dr. Jane Wu joining us, and she's been also a long-term supporter of the Les Turner Symposium. She has given talks in many times in the years, and it's very, I'm very happy to have her join us today. And she's also the new center member at the Les Turner ALS Center. Thank you so much, Dr. Wu. So the floor is yours. I'll stop sharing my slides.
Thank you very much for the kind introduction, and thank you and also organizing team for putting together this very exciting program. So today I will discuss about mitochondrial dysfunction in the spectrum disorder. And in case I run out of time, I would bring this important slide first. So I've been very fortunate to have a talented team over the years. Without their hard working, this talk would not be possible. I would not, their names will be on individual slides, so I won't go through here. We have a number of collaborators who kindly provided their valuable suggestions, shared their samples, reagents, and expertise. And we are grateful for the patients and the family and the funding sources. So frontotemporal lobe dementia, also known as frontotemporal lobar degeneration, is caused by neuronal loss in these brain regions, leading to executive dysfunction, cognitive impairment, changes in personality and behavior, and eventually succumb to the disease. While ALS is a motor neuron disease affecting upper or lower motor neurons, these two, somehow I cannot advance my slide. Can the tech team help somehow? Next slide, please.
Dr. Wu, you'll have to click on the slide to show a window.
I am in the slideshow window. It's somehow it's got frozen on me.
If you want to try to hit escape and maybe start the show again, but we don't have access.
I tried, so should I, so it's sharing. I tried to, I cannot do that. Okay, so now let me see, I still cannot advance slide.
Dr. Wu, I'll advance for you. Just let me know. Next slide.
Next slide, please. So these two seemingly distinct group of diseases, in fact, are overlapping significantly in clinical manifestation in genetics and in neuropathology. About 50 up to 50% of ALS patients also show cognitive impairments, while the FTD patients also show locomotor deficits. Genetic studies have reviewed more than 40 genes whose mutations cause ALS or FTD. I would like to draw your attention to a few of them. One is TART DNA binding protein, and a previous speaker has already alluded to. It encodes 43 kilodalton KD, 43 kilodalton protein TDP43. A landmark study by Norman Lee and Trojanowski in 2006 found that TDP43 is a protein component of the inclusion bodies, not only in ALS, but also in FTD. Now it's clear that more than 90% of ALS cases and about 50% of FTD cases have TDP43 pathology. In addition to TDP43, there are several other nuclear RNA binding proteins, including this gene called FUS or fused in sarcoma, HMP-A1, and others. Next slide, please. So FTLD can be classified into different subtypes according to the protein deposits in the patient samples, and we're going to focus on TDP43 and FUS. Next slide. So this just shows some of the various different forms of protein inclusions found in the cortex of FTD or ALS in their motor neuron. Next slide, please. My lab has longstanding interest in understanding the biological function of these RNA binding proteins in the nervous system, in particular, how mutations in these RBP genes or their dysregulation lead to neurodegenerative diseases, especially FTD and ALS. Today, I will share with you two short stories on the role of mitochondria in TDP-induced neurotoxicity. If time allows, I'll also go through some of the data on FUS proteinopathy. Next slide, please. Mitochondria are double membrane bound organelles. Their inner membrane is heavily folded to form these crystal structures. In this inner mitochondrial membrane are the respiratory complex, including complex 1, 2, 3, 4, and 5 are located. So complex 5 is also known as ATP synthesis. Mitochondria not only act as a powerhouse for energy metabolism, but also as signaling stations. Mitochondria are in active communication with the nucleus and the other subcellular organelles. Next slide, please. Both TDP43 and FUS are multifunctional RNA binding proteins. In the nucleus, they have multiple functions ranging from DNA stability to RNA splicing regulation. In the cytoplasm, they're important for translational control, stress granule formation, autophagy, and so on. In the mitochondria, they are multifaceted. I'm going to go into more details. So both of them are RNA binding proteins with RNA recognition motifs, and these low complexity domains with multiple functions. Multiple mutations have been identified as indicated by these vertical bars. I only highlight a few of them, which are well characterized, including this A315T or E mutant, and in FUS, the P525L mutant, which hits the C-terminus nuclear localization signal. Next slide, please. So first part is the role of the mitochondria in TDP neurotoxicity. Next slide. Next slide, please. Yes, to understand the role of TDP-43 in neurodegeneration in vivo, we generated transgenic fly models. In such models, we can target the expression of the human TDP-43 into subpopulations of neurons. For example, expression of the human TDP-43 in the Drosophila retina lead to age-dependent progressive retina degeneration, as shown here by both growth morphology and EM. Targeting TDP-43 in the brain regions responsible for learning memory lead to axonal degeneration. Next slide, please. Targeting TDP-43 in motor neurons lead to the motor neuron axonal degeneration, and including at the early stage axonal swelling, neuromuscular junction defects, accompanied by progressive loss of motor neuron function, and eventually motor neuron death. Next slide, please. So we also established stable cell lines, which express either wild type or mutant TDP-43 using biochemical fractionation and immunoelectron microscopy. Clearly, we show that TDP-43 is detected inside the mitochondria. Interestingly, when we examine the FTD TDP brain samples, it's clear that their mitochondria are significantly impacted. This panel shows a normal mitochondria look like with well-organized Christie structure. This is a cross-section. In the patient samples, mitochondria are severely affected with the Christie swelling and the degeneration of Christie, sometimes complete loss. Interestingly, in a subpopulation of mitochondria, these protein aggregates containing TDP-43 is also detected. Next slide, please. And such mitochondrial damage is also recapitulated in both cell lines as well as in fly neurons. This is normal mitochondria, and mitochondria are severely damaged with the swelling Christie, and the quantification show the significant increase of damaged mitochondria. Similar cases found in the motor neurons. Next slide, please. When we examine the different function of mitochondria, we found that in cells express either wild type or mutant TDP-43 membrane potential, mitochondrial membrane potential is significantly decreased, and with increased ROS production and decreased mitochondrial ATP synthesis. When we examine the different mitochondrial respiratory complexes, it's clear that complex 1 activity is severely compromised, to some extent also complex 4. Such mitochondrial dysfunction occur before any detectable cell death. Next slide, please. Because we found significant mitochondrial dysfunction and also protein aggregates inside the mitochondria, we asked whether TDP-43 affects this conserved pathway known as mitochondrial unfolded protein response, which is essential for maintaining homeostasis, proteostasis and mitochondrial health. Next slide, please. So when different markers of mitochondrial unfolded protein response, mitochondrial UPR, are examined clearly in both inducible cell model and the fly neurons, these genes are activated, including the mitochondrial chaperones and a mitochondrial protease known as LONG-P. Interestingly, in patient brain samples, there's also increase in the level of LONG-P in the brain samples. Next slide, please. So using biochemical assay and the in vitro assays, we show that purified mitochondrial protease LONG-P can the TDP-43 in cells when we downregulate LONG-P, which leads to the increase of mitochondrial level of TDP and decrease the cell survival. When we increase the LONG-P expression, that correlates to the decrease of cytotoxicity. Next slide, please. So similarly, downregulating the LONG-P homolog in a fly lead to the exacerbation of mitochondrial damage with concomitant increase of accumulation of mitochondrial level of TDP-43. And in these flies, locomotor neuron function is further exacerbated, leading to the decrease of locomotor index in both female flies and male flies. Next slide, please. Interestingly, when we overexpress two genes known for removal of damaged mitochondria, namely PINK1 or parking, mitochondrial damage is partially reversed with concomitant enhancement of locomotor function and increase the survival rate, extended the lifespan. Next slide, please. So together with studies from many groups, we propose this mitocentric view of TDP-43 induced neurotoxicity, in that expression of TDP-43 leads to various type of dysfunction in mitochondria, including the ATP synthesis and disrupted mitochondrial dynamics and transport, and inducing mitophagy or mitoautophagy. A recent study also suggests that such mitochondrial damage triggers the release of mitochondrial DNA and induce neuroinflammation. Next slide, please. So here's a brief summary of this part. Because of time constraint, I would just very quickly say that because genetically reducing damaged mitochondria resulting in improved neuronal function, increased lifespan, these suggest that mitochondrial dysfunction may be a key driver event in TDP-43 proteinopathy. Next slide, please. For the remaining few minutes, I'm going to very quickly go through the mitochondrial damage in FUS proteinopathy. Next slide, please. So similarly, we built the fly model for FUS proteinopathy. When FUS is expressed, we observed the neuromuscular junction defects and the locomotor function defects. I should mention that such fly models have been also generated by several other groups and with some similar findings. Next slide, please. So again, in motor neuron, FUS expression led to the mitochondrial fragmentation and the reduction in mitochondrial size. Next slide. So again, FUS is detected inside the mitochondria by the immuno-EM and disrupting the mitochondrial crystal structure. Next slide, please. So in these inducible cells, we can do the time course, which indicates that mitochondrial crystal damage and functional changes occur very early, way before the detection of cell death. Next slide, please. In this case, when we look at the different mitochondrial complex from 1 to 4, they are not affected, although mitochondrial ATP synthesis is clearly reduced. Next slide, please. In patient samples, we found not only increased FUS expression, but also severe mitochondrial damage. And interestingly, one of the mitochondrial chaperone genes called HSP60 is also increased. And we previously have shown that this chaperone mediates the translocation of FUS into mitochondria. Next slide, please. So when we look at the mitochondria complex 5, which is ATP synthesis, we found that the dimer formation and is compromised both in activity and at the level of the 5 dimer. So this suggests that the assembly of the mitochondria ATP synthesis is compromised by FUS. Next slide, please. Similarly, we asked whether mitochondrial unfolded protein response is affected by FUS expression at a different time course. You can see that in both cultured cells and in fly neurons, these mitochondrial UPR genes are induced. Next slide, please. So in the case of FUS proteinopathy, we examined the different mitochondrial UPR genes. We found that downregulating these mitochondrial UPR genes partially reverse the neurodegeneration phenotype, in this case, retina degeneration. And on the other side, downregulating of long P, which exacerbates TDP-43 neurotoxicity, this partially reverse the FUS neurodegeneration. Next slide, please. So in these flies, we also examined the genetic manipulation of the HSP60, which is, by the way, one of the mitochondrial unfolded protein response as well. In this case, when we downregulate by srRNA, and it partially reverse the mitochondrial fragmentation phenotype and it partially reverse the locomotor function defects in these flies. Next slide, please. So using biochemical assays, I will just very quickly summarize that not only FUS co-localized with ATP5B, which is a catalytic subunit of ATP synthesis in the mitochondria, biochemically they also interact with each other. Next slide, please. So together, our data led us to propose this working model for FUS-induced mitochondrial damage, in that either mutation or excessive expression of FUS lead to the mitochondrial translocation of FUS from the cytoplasm. Once inside the mitochondria, they interact with a catalytic subunit of ATP synthesis, causing perhaps a disassembly of mitochondrial ATP synthesis. And such disassembly triggers the mitochondrial unfolded protein response. Excessive induction or prolonged activation of mitochondrial UPR contributes to neuronal death. Next slide, please. So a large number of studies from other groups have also shown the various mitochondrial dysfunction, including different mechanisms such as sequestering the mRNA of the respiratory chain complexes and the calcium signaling and the disruption of ER mitochondria association. And more recent studies suggest that the mitochondria anchoring protein syntaphlin interacts with mitochondria may inhibit mitochondria transport. And the previous study suggesting the mouse model expressing FUS in motor neuron led to the mitochondrial damage before the detection of motor neuron loss. Next slide, please. To summarize, we show that expression of wild type of ALS mutant TDP43 or FUS led to mitochondrial defects, disrupting ATP synthesis, activating mitochondrial UPR, but they seem to act by different mechanisms. TDP interacts and degrades by mitochondrial protease, long P, whereas FUS directly targets the catalytic subunit of mitochondrial ATP synthesis, ATP5B. Our study suggests the role of nuclear RNA binding protein in mediating nuclear mitochondrial or mitochondrial nuclear communication. And I would like to emphasize the mitochondrial impairment is among the earliest detectable defects in FTD-ALS. Importantly, genetic manipulations that ameliorate mitochondrial defects also partially reverse these neurodegenerative phenotypes, suggesting that mitochondrial dysfunction is critical for pathogenesis. So these data suggest that reversing mitochondrial damage, restoring mitochondrial function may provide therapeutic benefits for FTD-ALS patients. Next slide, please. So we would like to propose that balanced nuclear mitochondrial communication is crucial for cellular cell health and for mitochondrial health and the cell health, and various mechanisms from early studies. The misfolded protein would cause mitochondrial impairment, including not only FUS, but previously demonstrated in the case of SOD and other genes associated with FTD-ALS. More recent studies show that there's another gene, C9orf72, it's itself encode a protein associated with inner mitochondrial membrane and critical for complex 1 assembly. Whereas the polynucleotide repeats translated the poly-GR also impair mitochondrial function. And all these together would disrupt mitochondria and nuclear communication. Next slide, please. And various pathogenic mechanisms have been implicated in FTD-ALS among these. And in red stars are those functions directly related to mitochondria. And I would like to propose that there are emerging evidence that these other domains are also indirectly associated with mitochondrial signaling. So with that, next slide, please. I would like to thank you all for your attention and be happy to entertain questions.
So maybe I should remind people to write their questions in the question and answer box. And maybe I should start with the questions that have already been asked. One of the participants asked, what are your thoughts on the impact of statins on mitochondrial dysfunction?
I didn't hear yours last.
What are your thoughts on the impact of statins? Impact of statins, yeah.
And I think that there have been some mixed conflicting reports. So it's a very complicated story. It affects various pathways, including some of the mitochondria related pathway. But at this point, I would say that studies are, I would say, conflicting.
Okay.
There's not overall clear indication one way or another.
All right, wonderful. So another question is, can you see any aggregation of FUS or FUS on mitochondria, or do they have an impact on mitochondrial organization?
Clearly, FUS has overexpression, either the wild type overexpression or ALS mutant to compromise mitochondrial function. So far, we have not detected similar to TDP inside the mitochondria, the aggregates. And in cell culture, in the cytoplasm, you can see the accumulation of the protein inclusion bodies. And this probably also related to the fact that there's liquid phase transition features are different between the two proteins.
Okay, so one more question. What happens to ROS production with either FUS or TDP overexpression?
Interestingly, so in the case of FUS, very early on, there's seem to be an early increase. Well, in both, I would say in both. I meant to say that in the first case, early on, there's a membrane potential in early stage, but later reduction. So in both TDP-43 and the FUS, mitochondrial ALS expression are increased. So that also contributes to the mitochondrial damage.
Very nice, yeah. So one more question. It says, I wonder how TDP-43 and FUS get imported into the mitochondria, and if inhibition of the importing process could rescue these disease phenotypes.
So that has been partially addressed by several groups, including Xinglong Wang's groups and the Seth Masters groups. And so there, TDP-43 seem to be imported through the TIM-TOM complex, and the evidence for FUS is less clear. And in the case of FUS, we show that mitochondrial chaperone, HSP60, plays an important role. That has been published. I can send you the paper. So the chaperone mediates the translocation of FUS into mitochondria. So the detailed pathways still need to be worked out.
Yes. So one more question from Dr. Pioro. Could upregulating long P expression be a potential therapeutic to reduce TDP-43 mitochondrial toxicity?
That's a great question. And we, in fact, have tried that. And unfortunately, just increasing the long P expression at a high level by itself perhaps affect many other aspects of mitochondrial function. And also long P has a large number of substrates. So just increased long P expression actually lead to more toxicity by itself. So we could not detect or could not examine whether TDP specific role there. So maybe more research is necessary to dissect out the pathway specific or gene specific effect. Or, so that is why all these are interconnected. Many of these proteins, including LAMP, have multiple partners. So it's not a simple one-to-one connection.
All right, so maybe I can have a short question. So Dr. Wu, you have shown very nicely that the inner mitochondrial membrane is broken within the context of TDP and also FAST. You have shown it in the human samples. And we have shown it within the context of ALS with upper motor neurons as well. And it seems as if like being able to repair that inner membrane of mitochondria could be a therapeutic target, because that's very important for the generation of ATP and for mitochondrial function. So, and we, you know, Dr. Colin Franz also mentioned about this, you know, personalized medicine approaches. So if you put these two together, do you foresee that a therapeutic intervention, mainly, you know, focusing on mitochondrial health, and would it be possible to identify patients who develop the disease due to mitochondrial defects? And you have even shown, you know, therapeutic interventions at a cellular level, at an animal model level. Do you foresee a future for this, for clinical trials and for treatment strategies?
I believe that Dr. Silverman would talk about those drugs which also seem to enhance mitochondrial health, and whether they specifically target the inner mitochondrial membrane, whether they are specific to compounds or few specific genes which can specifically repair the inner membrane, remains to be investigated. Yes, indeed, repairing the damaged mitochondria is clearly important, and how to specifically impair the inner membrane, that's interesting topic for further study. Okay,
so I have two quick questions. One is from Dr. Uday Pandey. He asks, does FUS localize to mitochondria in neurons only, or is it a non-cell type specific event?
I don't think it's a cell type specific, because in non-neuronal cells that can also occur. And remember, all these ALS genes, the vast majority, I would say, if not all, are expressed in almost all cell types. And they are studies suggesting that expression of TDP or FUS in non-neuronal cell types may also impact on the neuronal health in the nervous system. For example, glial cells and astrocytes, and they all express FUS and TDP.
All right, thank you so much. So one last question, Dr. Valerie Kilman asks, can you elaborate why down-regulating of UPR might help rather than up-regulating if there's a protein aggregation problem?
And that's a great question. That shows the complexity of this process. So interestingly, in other studies, for example, in worm and in others, sometimes downregulating UPR and even downregulating mitochondrial proteins would provide the benefits for the long-term health. So sometimes they are put into overdrive. So we think that prolonged excessive UPR could also damage. So this has been the case both in cell models and animal models as well. So this there's early and early repairing effects, or trying to set back to more normal condition, but prolonged excessive activation would cause detrimental effects to cell health. That is why balanced nuclear mitochondria or mitochondrial nuclear communication is so crucial for resetting the thermostat and then helping to maintain the cellular and organismal health.
Thank you so much, Dr. Wu.